Right now, inside your body, there's a secret you have never read. It is written in a language 4 letters long, copied into nearly every one of your 37 trillion cells, and yet almost no one alive has ever seen the whole of it. Somewhere in those 3 billion letters, there could be a variant so rare that fewer than 1 in 100,000 people on Earth carry it. You could be one of them right now, reading this sentence completely unaware. Maybe it is why you sneeze when you first step into sunlight. Maybe it is why certain foods taste like soap to you, while everyone else at the table swears they are delicious. Maybe it is something far stranger, something that has never once been explained to you, hiding quietly in a molecule you will never see, waiting for the right scientist, the right test, the right question to reveal it. This is the story of that hidden code, the story of the traits so rare, so strange, and so specific that they blur the line between medical mystery and evolutionary miracle. And by the end, you may find yourself looking at your own hands, your own reflection, wondering exactly what your DNA has been keeping from you. To understand and how something this small can hide something this significant, you have to go back to the molecule itself. DNA is not some abstract scientific concept locked away in a laboratory. It is a physical thing, a double helix twisting like a spiral staircase, so long that if you uncoiled the DNA in a single human cell, it would stretch to nearly 6 feet. Now multiply that by the trillions of cells in your body, and you get a length of genetic material that could stretch to the sun and back several times over. And yet all of that information is compressed into a nucleus so small that thousands of them could fit on the head of a pin. Inside that compressed spiral sit the 4 chemical bases, adenine, thymine, guanine, and cytosine, pairing with each other in a code so elegant that every living thing on this planet, from the bacteria in your gut to the giant sequoias in California, uses the exact same alphabet to write the software of life. What differs from person to person, from species to species, is not the alphabet, it is the sentence. And somewhere in your sentence, there might be a typo so unusual that geneticists would want to study it. Most of what makes you biologically unique comes down to variation. Small differences in the genetic code that shift a protein here, alter an enzyme there, change the shape of a receptor just slightly enough to produce something the rest of the population never experiences. Scientists call these variants polymorphisms when they are common and mutations when they are rare. But rare does not mean insignificant. In fact, some of the rarest genetic variants in the human population turn out to reveal the most about how our bodies actually work, because they expose what happens when a single gene behaves just a little differently than it does in almost everyone else. And this is where the story gets personal, because chances are, without knowing it, you already carry at least one. Consider something as simple as sneezing. For most of human history, no one thought twice about why some people sneeze, sometimes 2, 3, even 4 times in a row, the instant they step out of a dark room into bright sunlight. It seems too strange to be biological, more like a quirky habit than a genetic trait. But researchers eventually discovered that this reaction, called the photic sneeze reflex, is real, is measurable, and is inherited. Somewhere between 18 and 35% of the population experiences it, meaning it's not vanishingly rare, but it is rare enough that most people have never heard of it, and even rarer, is a true scientific consensus on exactly why it happens. The leading theory involves a kind of neurological cross-wiring. The optic nerve, when suddenly flooded with light, sends a signal so strong that it spills over into the trigeminal nerve, the same nerve responsible for triggering a sneeze. Your brain, in other words, briefly confuses a burst of light for a nose full of pepper. If you've ever wondered why sunlight makes you sneeze, know this. You inherited that reflex from someone, and they inherited it from someone before them. A slightly miswired shortcut in the nervous system passed down like a family heirloom, hidden in plain sight for generations. But the photic sneeze reflex is only the beginning. Beneath the surface of what looks like ordinary human variation lies a vast library of traits so specific that entire scientific papers have been written about a single family, sometimes a single person. Take asparagus. For most of history, no one questioned why, after eating asparagus, some people notice a distinct sulfurous smell in their urine within minutes, while others notice nothing at all. It turns out this isn't really about who produces the smell. Nearly everyone's body breaks down the sulfur compounds in asparagus the same way. The real divide is in who can smell it. A specific genetic variant near a cluster of olfactory receptor genes determines whether your NE is sensitive enough to detect those compounds at all. Some studies suggest roughly 1 in 4 people simply cannot smell it, no matter how close they get. It is a trait hiding not in your body's chemistry, but in your ability to perceive your own body's chemistry, a genetic filter on reality itself, and you've likely never once stopped to wonder which side of that filter you're on. And once you start looking at the sense of taste, the rabbit hole goes even deeper. There is a well-documented compound called phenylthiocarbamide, or PTC, that tastes intensely, unbearably bitter to some people, and completely tasteless to others. It comes down to a gene called TAS2R38, and the variation is so binary that scientists have used PTC paper strips as a genetic test since the 1930s, decades before DNA sequencing even existed. If you are a taster, a molecule most people can't detect at all might feel like biting into pure poison. If you are a non-taster, you could chew the same strip and shrug. This single gene may explain why certain vegetables, broccoli, Brussels, sprouts, kale, taste offensively bitter to some people and perfectly pleasant to others. It is not pickiness, it is not a preference. It is a hardware difference in the sensory receptors on your tongue, dictated by a few letters of code you never chose and never knew you were reading. Then there are the traits that go beyond sensation and into something almost architectural, differences etched not into perception, but into the very structure of the body. Heterochromia, the condition where a person's eyes are two different colors, or where a single iris contains two distinct colors within it, occurs in a small fraction of the population, often less than 1% when it isn't caused by injury or disease. It results from uneven melanin distribution during development, sometimes triggered by a genetic mutation, sometimes by a mosaic pattern of cells that never fully synchronized while the eye was forming. For most of human history, heterochromia was treated as a mark of the supernatural, associated with witches, wolves, mystics, and myths. Today, geneticists understand it as a beautiful accident of pigment cell migration, a visible reminder that even something as seemingly fixed as eye color is the result of a delicate, occasionally imperfect dance of cellular instructions during the earliest weeks of life in the womb. And speaking of cells that never fully synchronized, there is a phenomenon so strange that when it was first confirmed in humans, it nearly rewrote the rules of genetic identity altogether. It's called chimerism, and it means that a single person can carry two entirely different sets of DNA in their body at once. Not similar DNA, not related DNA, two genetically distinct genomes coexisting inside one human being, often without a single outward sign. It typically happens when twin embryos merge early in development, one absorbing the other, so that a person can end up with skin cells carrying one genetic code and blood cells carrying another. In extremely rare and now well-documented cases, this has led to almost unbelievable legal situations. Mothers who failed maternity DNA tests because the genetic material in their blood didn't match the genetic material in their eggs, simply because they were their own twins' genetic inheritor as well as their own. Somewhere on Earth right now, there are people walking around chimeric, carrying a biological ghost of a sibling who never fully became a separate person, and many of them will live their entire lives never finding out. This raises an uncomfortable, thrilling question. How many rare traits are quietly written into your own body, doing nothing, causing no symptoms, waiting for a scientific reason to be found? For most of human history, the answer to that question was permanently unknowable. Genetics were something you inherited blindly, expressed silently, and passed on without ever fully understanding. That changed only recently, and the story of how it changed is itself one of science's greatest underdog tales. In the early 1950s, the structure of DNA itself was still a mystery being pieced together in competing laboratories across the world. It took the X-ray crystallography work of Rosalind Franklin, whose famous photograph 51 revealed the unmistakable X-shaped diffraction pattern of a helix, combined with the model building insight of James Watson and Francis Crick, to finally reveal that DNA was a double helix, two strands twisted around each other, connected by base pairs in a structure so elegant it practically explained its own function. Franklin's critical contribution was, for decades, underacknowledged, a quiet injustice in the history of one of biology's greatest breakthroughs, only properly recognized by history much later. But once the structure was known, an explosion of understanding followed. Scientists realized that if DNA was the code, then somewhere in that code had to be the instructions for everything. Eye color, disease risk, ancient ancestry, and yes, the rarest traits imaginable. But knowing DNA had a structure was very different from being able to read what it said. For nearly 50 years after Watson, Crick, and Franklin's discovery, scientists could see the shape of the code but not its content, like holding a book written in a language no one could translate. That changed with the launch of the Human Genome Project in 1990, an international multi-billion-dollar effort to sequence all 3 billion base pairs of human DNA, letter by letter. It was, at the time, one of the most ambitious scientific undertakings in history, involving thousands of researchers across multiple countries, racing against time, against funding constraints, and eventually against a private competitor determined to beat the public effort to the finish line. When a working draft was finally announced in the year 2000, and a more complete sequence followed in 2003, it wasn't just a technical milestone, It was the moment humanity, for the first time, could actually begin to read its own source code. And what that source code revealed was staggering. Humans share more than 99% of their DNA with every other human on Earth. The vast diversity of human traits, appearances, and rare conditions comes down to differences in a tiny fraction of the genome, meaning the rarest traits in existence are hiding in some of the smallest cracks of genetic variation imaginable. Once the human genome could be read, scientists could finally start connecting specific traits to specific locations in the code, and that's when some of the strangest human variations in history began to make biological sense. Take the case of a family identified by researchers as having an unusually low sensitivity to pain, so extreme that some members could place their hands on hot surfaces or suffer serious injuries without registering typical pain signals. Investigations eventually traced this back to mutations affecting sodium channels in pain, sensing neurons, specifically a gene called SCN9A. In people with certain rare mutations in this gene, pain signals simply never fire correctly, no matter how severe the injury. It sounds almost like a superpower, and in some ways it has inspired serious medical research. Because if scientists can understand exactly how this gene blocks pain, they may one day be able to develop entirely new classes of painkillers, modeled after nature's own rare accidental discovery. But it is also a double-edged trait because pain, for all its unpleasantness, exists for a reason. It's your body's alarm system. People with this condition often suffer repeated injuries specifically because they never receive the warning signal the rest of us take for granted. A haunting reminder that even the traits that sound miraculous often carry a hidden cost. The virus depends on to get inside. This discovery became one of the most significant in modern medicine, directly informing groundbreaking bone marrow transplant treatments that have, in a small number of documented cases, resulted in patients being functionally cured of HIV after receiving stem cells from donors carrying this rare mutation. It is a stunning example of how a single, rare genetic quirk, likely originating and spreading through European populations for reasons still debated among scientists, some theories point to historical plague resistance, can end up reshaping the future of medicine centuries later. And this pattern repeats again and again throughout genetic history. Rare traits that seem like biological anomalies often turn out to be evolutionary strategies, quietly preserved in the population because, under the right circumstances, they offered a survival advantage. Consider sickle cell trait, which is far more common in populations with historical ties to regions where malaria was endemic. Carrying a single copy of the sickle cell gene mutation, not two, which causes sickle cell disease, but one, can provide significant resistance to malaria infection because the parasite struggles to thrive in the misshapen red blood cells the mutation produces. It is a genetic trade-off written directly into the population's survival history, evidence that evolution doesn't create perfect solutions, only workable ones, and that even a trait with real medical complexity can exist because generations ago, it kept someone's ancestors alive long enough to have children. The story of lactase persistence follows a similar arc, though it unfolded on a different timeline and for different reasons. For most of human history, and still today for a majority of the world's adult population, the ability to digest lactose, the sugar found in milk, disappears after childhood. This is, biologically speaking, the default human condition. Infants produce the enzyme lactase to digest their mother's milk, and then, once weaned, that production naturally shuts down. But somewhere around 10,000 years ago, in populations that began domesticating cattle and relying on dairy as a food source, a genetic mutation emerged that kept lactase production switched on into adulthood. This mutation spread rapidly through certain populations, particularly in Northern Europe, not because it was rare and special, but because it was wildly advantageous, offering a reliable new food source in a world where famine could be a death sentence. Today, lactase persistence is common in some populations and genuinely rare in others, making it one of the clearest examples in human genetics of natural selection, acting on a single, identifiable mutation within a remarkably short evolutionary timeframe. It is, in a very real sense, evolution caught in the act, still visible in the genetic code of billions of people alive right now. But not every rare trait comes from ancient evolutionary pressure. Some come from something far more recent, far more experimental, and far more controversial. The moment scientists stopped simply reading the genetic code and started learning how to rewrite it. This shift began quietly, with research into bacterial immune systems in the early 2000s, when scientists studying how bacteria defend themselves against viral infections noticed strange, repeating genetic sequences in bacterial DNA, interspersed with fragments of viral genetic material the bacteria had previously encountered. These sequences were named CRISPR, clustered regularly interspaced short palindromic repeats, and for years they remained a biological curiosity, interesting but not obviously useful. That changed dramatically when researchers, including Jennifer Doudna and Emmanuel Charpentier, realized that this bacterial defense system could be repurposed into a programmable tool, one capable of cutting DNA at a precise, chosen location, almost like a pair of molecular scissors guided by GPS coordinates written in genetic code. The implications were immediate and staggering. For the first time in human history, scientists had a relatively simple, affordable, and precise way to edit the genome of any living organism, including, eventually, human cells themselves. This same technology, ancient DNA extraction and sequencing, has also transformed an entirely different field, forensic science. For most of the 20th century, solving crimes or identifying remains relied on fingerprints, dental records, or eyewitness testimony, all methods with serious limitations. That changed permanently with the development of DNA fingerprinting in the 1980s by British geneticist Alec Jeffreys, who discovered that certain repetitive sequences in human DNA varied enough between individuals to create what was, essentially, a genetic identification code unique to each person, aside from identical twins. The technique was first used forensically in 1986 in a case that both convicted a guilty man and exonerated an innocent one who had already confessed under police pressure, a chilling reminder of how badly the world needed a more reliable way to separate truth from coincidence. Since then, DNA forensics has evolved into an extraordinarily sensitive science, capable of identifying individuals from samples so minuscule they would have been useless just decades earlier. A single hair follicle, a trace of saliva, sometimes even skin cells transferred through nothing more than a brief touch. It has overturned wrongful convictions decades after the fact, identified remains from mass disasters and historical mysteries, and reunited families separated for generations, all because the rarest, most specific sequences hiding in a person's DNA turned out to be as unique and identifying as anything humanity has ever discovered. And this brings us back to you, and to the question that has been quietly building beneath every discovery in this story. If DNA can reveal ancient interbreeding with extinct human species, if it can expose hidden immune advantages, silent chimeric twins, and painless nerve mutations, if it can be edited, sequenced, and traced across tens of thousands of years, then what exactly is still hiding in yours? Perhaps it's something as small and strange as being unable to smell asparagus in your own urine, a trait so minor it barely registers as a mystery. Perhaps it's something structural, like a faint mosaic pattern in your skin. Pigmentation you've never noticed because no one has ever looked closely enough to see it. Perhaps it's ancestral, a fragment of Neanderthal or Denisovan code, quietly shaping something about your immune response or your pain tolerance in ways modern medicine is only beginning to map. Or perhaps it's something science hasn't even identified yet. A variant so rare it exists in only a handful of documented people worldwide, waiting for a genetic sequencer, a curious researcher, or a strange coincidence to finally bring it to light. What makes this field so genuinely thrilling isn't just the traits already discovered, it's the undeniable mathematical certainty that more are still out there, hidden in plain sight. Inside ordinary people, living ordinary lives, completely unaware that their bodies contain something worth studying. Genome sequencing, once a billion-dollar, decade-long international undertaking, can now be done for a fraction of that cost in a matter of days, and as more and more people around the world have their DNA read, letter by letter, researchers continue to stumble across variations so unusual they redefine what scientists thought they understood about human biology. Every rare trait discovered so far, the painless mutations, the malaria resistance, the ancient hybrid ancestry, the twin-absorbed chimeras, the mosaic skin patterns, the sensory quirks passed down like secret family heirlooms, was at some point simply sitting quietly inside a person who had absolutely no idea it was there, until science finally asked the right question. And that is perhaps the most extraordinary truth revealed by decades of genetic discovery. DNA is not a fixed, finished story you were simply handed at birth. It is closer to an ancient manuscript, endlessly copied, endlessly rewritten across billions of years, filled with the fingerprints of extinct species, evolutionary trade-offs, silent mutations, and rare accidents of biology, all folded into a molecule so small it is invisible to the naked eye, yet powerful enough to shape everything from how you taste your food to how you survive a virus to whether sunlight makes you sneeze. Every single person alive is walking around as a living archive of billions of years of trial, error, survival, and chance, carrying quite possibly a genetic secret unique enough to be worth discovering. The only question left is whether you will ever find out what yours is, or whether it will simply remain there, quietly written into every cell of your body, the way it has been all along, waiting patiently in a language you were never quite able to read.